So we have the first speaker today. He's Arnold Abakah from Ghana, and he's going to talk about conformational dynamics and active site flexibility in the Pseudomonas aeruginosa, LasR protein inhibiting the quorum sensing receptor. Welcome, Arnold, and please proceed to the presentation.
Thank you, Sarah. Thank you, everybody. Hi, everyone. I hope you're all good. I'm very excited to be presenting to you all today. I'm presenting on disrupting quorum sensing. Today's talk is going to be about a lot about quorum sensing in Pseudomonas aeruginosa. And in our experiment, we used computational methods to explore the conformational dynamics and active site flexibility of what is called the LasR protein. But before I begin, I want to just say a little bit about myself. I'm currently a research assistant at the Quorum lab in KNUST and KCCR in Kumasi, Ghana. My interest is in the omics of pathogens. And when I'm really not in the lab, I enjoy community outreach. In fact, I was even from one of these outreaches here. I had to pause and come for the conference. You find me singing or discovering world cuisine. So this work was carried out in KNUST, and KNUST is actually really excited to be at BSI this year. KNUST has a very lush and green and serene environment, and recently we were named or we were ranked as #1 in the global provision of quality education by Times Higher Education. So the background to our work is in lymphatic filariasis, or mostly called elephantiasis. And this is a debilitating disease that has been pointed out by WHO as a very critical NCD in the tropics, right? And it has been estimated by WHO to affect about over 120 million globally people. What we have done with over 20 years of experience in this field is really characterize the host response and immunity of these patients. And what we've come across, or what we've really found recently, is that these lymphatic filariasis wounds usually end up with end up with chronic non-healing wounds that are very debilitating, and it increases their morbidity and mortality. And these wounds usually end up as chronic because of the secondary bacterial infections that are present. Following is a very sensitive content that might be sensitive to some of us, but this is the pathology of the disease. It disrupts or damages the lymphatic system, which causes drainage or fluid accumulation in the lymphatic systems, resulting in swelling of the legs, and sometimes resulting, or most times resulting in wounds. And these wounds become pathogenic because of the proliferation or infestation of secondary bacterial infections in them. A lot of work has been done, as I said earlier. We have really characterized these wounds and found that most of the secondary bacterial infections in there are as a result of the colonization of multi-drug resistant bacteria in these wounds. And one of the most prominent of the bacteria that are present or that have been found through our characterization methods is Pseudomonas aeruginosa. And so it was very interesting to us that when we found out that there has been work done in this field, right? Pseudomonas aeruginosa is essentially a gram-negative pathogen or gram-negative bacteria that infects immunocompromised patients. And LF patients happen to fall within this bracket of patients. It proliferates, or its pathogenicity is as a result of its ability to form biofilms. What biofilms are is really a surface coating that prevents the treatment of antibacterial or antimicrobial substances on them. And so, for example, it has been noted that when biofilms are formed, especially on medical devices in the hospitals, even the harsh environment or the harsh conditions of chlorine is unable to clear these bacteria because they possess the these biofilms that protect them. Now, one of the important proteins in this process, or in this process of biofilm formation, is called LasR. And what LasR is really a transcriptional activator of the LAX system of Pseudomonas aeruginosa. So the LAX system has been found to be related to the bacterial biofilm formation in Pseudomonas aeruginosa. And how this works is that when the bacterial population reaches a specific density or a specific number of bacteria are aggregated at the site of infection, for example, in these wounds, it senses the presence of its autoinducer, which is produced by the bacteria normally. And with the right amount or with the right population of bacteria, the neighboring bacteria sense it, so the autoinducer enters the, let me try to use the pointer. So this is the autoinducer. For example, it attaches to the LasR protein, which is the transcriptional activator. And upon attachment, it continues to transcribe the genes that are responsible for the bacterial biofilm formation, for example. And our interest was really how we could target these LasR proteins because they are very essential in the bacterial biofilm formation, right? So as I've already explained, LasR is important in the virulence and biofilm formation of bacteria. It enables communication among these bacteria and incorporates or strengthens the infection persistence. When we sought out to see how we could target biofilm formation in Pseudomonas aeruginosa, we were fortunate to come across the work done by Wang and his colleagues, where they tried to use cladodionin, which is a natural product from the marine fungus Cladosporium species, right? Now, what they did was that they did several in vitro studies that had to use biofilm, bacterial biofilm assays to study how cladodionin affects biofilm formation. And they had very promising results. They also went ahead to do some docking studies with the cladodionin, and they realized that it was an inhibitor for the LasR and the PKSR receptors, which are both essential receptors in P. aeruginosa biofilm formation. Now, what we really did here in our study was that we picked CLA-O, another ortholog of cladodionin, and bound it to the active site of the LasR protein. So we were really looking into the chemistry associated with this biofilm formation activity of cladodionin. And this is really good news for us because if you are able to target the biofilm formation system or the biofilm formation machinery in P. aeruginosa, it means that our patients are going to have some success in the treatment of their wounds. Now, the study was really or solely computational. We had to get our proteins from the RCSB database, prepare it by removing heteroatoms and water molecules, for example, and then we also retrieved our ligand, which in this case will be the autoinducer called AHL or 3-oxododecanoyl homoserine lactone. So from now, I'm going to call it AHL. So when you hear AHL, it means that is the autoinducer. And we also prepared the CLA-O as from the online database. We then did docking studies with both the CLA-O with the LasR protein and the AHL with the LasR protein. We went ahead to do molecular dynamic simulation over 200 nanosecond time scale, and then we did some post-molecular dynamic simulation analysis. We sought to see what actually was going on with the interaction between CLA-O, which is our marine fungus-derived secondary metabolites, that is our potential drug in this study. And we did computational studies with it, and the docking results showed several interactions with the active site residues. So on the left, we have the AHL, which is the native ligand or the autoinducer, that starts or causes the bacterial biofilm formation machinery to be assembled. And on the right, we have the potential therapeutic target, which is CLA-O. And we see the interactions quite clearly. We see that it's the CLA-O. In the CLA-O, there's interactions with hydrophobic and hydrophilic amino acid residues, as well as some water molecule interactions. We went ahead to see how exactly the stability between the binding of the LasR protein with the CLA-O was carried out across the 200 nanosecond timescale. And we saw that, just a minute. Yes, we saw that in red, we have the AHL bound or native ligand bound to LasR, and in green, we have the CLA-O, and in black is the apoprotein or the LasR protein, which has not been bound. And we see that the CLA-O causes stable or exhibits a stable binding throughout the reaction until it gets to about 150 nanoseconds, where it significantly drops, right? And while, as expected, the AHL or the native ligands exhibited a stable binding throughout. And so we see that CLA-O, although stabilizes the protein, but after some time, the stability reduces as compared with the AHL. We went ahead to do the radius of gyration, which is a measure of the flexibility of the binding. We see that as with the AHL bound protein, we see that there is significant flexibility. However, the spikes in this case represent the mobility or the distance or the distances moved in the case of flexibility. So what I'm trying to say is that when we bind the ligand with the protein, we are expecting it to bind compactly, but at the same time, there should be some stability or flexibility. And we see significant motility with the AHL, which has high sparks than the CLA-O bound ones, right? Which means that CLA-O binds compactly, right? Which can be the cause of its inhibitory effect with the LasR protein. And the radial distribution function also confirms this. We see that with the apoprotein, there is binding, there is some flexibility, so the protein is still able to move. But with the CLA-O bound state, we see that there is little flexibility, which tapers at the end. Now I went ahead to see how the residues or the parts or the amino acid residues in the protein behaved when we bound them. We see, as I repeat, that the CLA-O is the green and the AHL bound state is the red. We use the apoprotein as the standard because although we are not expecting to see much changes, but we used it as a standard. We see that the AHL state exhibited fluctuations in some of the residues, right? But the CLA-O at the end, for example, after about 170, it's in this region, we have the amino acid 170 to about 180, right? And we see very high spikes, which means that there were fluctuations in the 170 residue. And this 170 residue, according to literature, has been termed or is called the L3 loop. And we know that the L3 loop is important in the binding efficiency or in the function of the protein as a DNA binding protein, right? So if we are inducing fluctuations in this loop, then it means that the binding of the LasR protein with the DNA to cause the transcription is being impaired. And this is good news. We also went ahead to see the number of contacts with the active site residues in the active site of the protein. And we see that the CLA-O has higher spikes, which means that it had a lot of contact. So throughout the simulation, we see that with the residue 1, there's a contact, very high contact. After about 170, we also see a very high spike, and throughout it's consistent. We went ahead to see we extracted the frames or across the 200 nanosecond scale. In A, we have the 50 nanoseconds. In B, we have the 100 nanoseconds. In C, we have the 200 nanoseconds. And we saw that CLA-O was binding to specific amino acids, right? And what was interesting over here was that we saw CLA-O, even though had interactions or hydrophobic interactions with some of the residues at the active site, we also see that it has interactions with water molecules at the active site. What this suggests is that because of the flexibility, as we mentioned earlier, the water molecules were able to infiltrate the active site, right? We do not necessarily, which means that it destabilizes the binding. And this is what we want to achieve, right? We want it to bind perfectly first, but then again, we don't want it to bind too tight, or we want the binding to disrupt the whole system so that the LasR system is disrupted, and then we achieve our anti-inhibitory or the quorum sensing inhibitory effect. So what we've done so far in our work is that we targeted the LasR protein of Pseudomonas aeruginosa, which is a very critically pathogenic pathogen, right? Or critically important pathogen in the chronicity of wounds in our case, right, with LF patients. And we use computational analysis to see or to examine the binding analysis and the flexibility assessment, right, or the binding nature or the mechanisms going on with the binding of CLA-O, which is our potential drug or potential quorum sensing inhibitor drug, right, with the LasR protein, which is a very important protein in quorum sensing. Now, what I get asked most often that does this mean that we could use CLA-O to treat Pseudomonas infections off the top? Currently, there's not much work being done in that area, but because of this binding affinity with LasR, we are hypothesizing that we can use CLA-O in combination with several other antibiotics, for example, or the last line of antibiotics, because Pseudomonas aeruginosa has actually been found to be resistant to the last line of antibiotics. So we use the CLA-O in this case to bind or to disrupt the quorum sensing ability of Pseudomonas aeruginosa. And then the antibiotics come in later on to, kill the bacteria outright. So this is the future of the natural product of the use of natural product against antibiotic resistance. And it is very important for us to notice that. I would like to acknowledge my co-authors, Prince, Pakwesi, Priscilla, and Dr. Ales for their help with this project. Thank you very much.